this is the third case study, which is carbohydrate vaccines, and it's a nice topic because it brings in some of the issues we've been discussing before in terms of glycan sequences, but it also poses some really nice challenges in what we can say is biomolecular technology in terms of the design of vaccines. Carbohydrate-based vaccines, although they're effective, they're not easy to characterize, and there can be problems for the manufacturers in terms of specifying the character of the inherent polydispersity of these types of molecules compared to protein-based vaccines. But these vaccines are available now and have been available for some years. I think this Menactra vaccine came out nearly 20 years ago from Aventis, now part of Sanofi. And one of the organisers last week was from Sanofi. That was Bernardo. But Sanofi are not the only companies that are producing these vaccines. Merck were very strong on this, and then GSK with their plant in Belgium at Rixensart are producing their own vaccines against serious disease like meningitis. Okay, so vaccines. Vaccine produces immunity. So we've all been vaccinated against COVID. Tony and I have had 4 doses. I think I don't know how many. You probably all had 3, I would guess. So that's just an example. Because Tony and I are older, we've also had flu vaccines. I guess you guys don't have the flu vaccination. So vaccine against influenza. So a vaccine produces immunity, and the response is similar to natural infection, but without the risk of disease. So I unfortunately caught COVID. Fortunately, not the early 2 years ago, but the latest lot, which Omicron, which are not so nasty. And because I've been vaccinated before, it meant that the seriousness of the infection was not great. But having caught COVID, I also developed resistance antibodies as a consequence of that. So I would think I'm quite robust for jabs and having got the antibodies from the infection. So the response from vaccination is similar to natural infections, but without the risk of disease. And certain bacteria with encapsulating polysaccharides are particularly dangerous. It's not all bacteria have these capsular polysaccharides, but some do, and some of the dangerous bacteria do. So we can take advantage of that in targeting the polysaccharides with the generation of our vaccine. So we design a vaccine based on the polysaccharide. Now, I don't know whether you can see this. Maybe you could turn the lights. They'll have no effect, of course, on the recording, but you can probably see these are bacteria here. Oh gosh, let's go back. And you can just see this outline, see this boundary of this film or capsule of polysaccharide or glycocalyx surrounding the bacteria. This one here actually is in the process of cell division by the looks of it. But this is the stuff which we are interested in. And see, these are some of the dangerous capsular bacteria. Not all bacteria are dangerous. Not all capsular bacteria are dangerous, but these are some of the bad ones. So you've got Streptococcus pneumoniae. You've got Group B Streptococcus, Neisseria meningitis or meningococcus, and Haemophilus influenzae. So these are all bacteria which produce disease and have these capsules of high molecular weight polysaccharides, and these are relatively complex polysaccharides. They're not simple single chain ones like like amylose and cellulose, but they can be quite complex with branches and several types of saccharide residue here. So these are a given species can have different polysaccharides, or we call these serotypes. So different types. A given species, like Group B Streptococcus, can have several different types of what we call serotypes, depending on the nature of the capsular polysaccharides present. And we did this, actually showed this a few weeks ago, and asked, it was you, I think, got it, Peter, could you spot the difference between these two serotypes in terms of the saccharide sequence? You've got glucose, lactose, and acetylglucosamine, and N-acetylneurominic acid, sialic acid. And by the end of the MRes course, you'll know all these sequences, of course. Yeah, that's that. And you should know them now, really. By the way, we can lend you copies, and you can't see this on recording, apologies, of this book, Introduction to Polysaccharide Biotechnology. This has been superseded by the second edition of this, which has several other authors joining us. So we've got several copies of the older version, which is still good, which we can loan out if you want to borrow these. But in here in chapter 1 is a nice table giving the sequences. I think I also may have showed that in the very first introductory lecture for this module. I gave you something on glycan types, I think. I gave you a table in there. But yeah, chapter 1 here is quite quite useful. So you can see they differ subtly in terms of the type of link. This is a 1,4 link here, and this is a 1,3 link, beta 1,3 link, beta 1,4 link here. Okay, and these are other serotypes of Group B. So we just looked at group serotype 1A and 1B. You can see there's more complex forms, but all with certain characteristic residues, such as this one, sialic acid, and N-acetylneurominic acid, but the sequences are different. And that's important when designing, vaccines against these subtle changes in sequence can be important. These are capsular polysaccharides from serotypes of N meningitis, Neisseria meningitis, and again you can see they are quite different. Actually, these are a linear unbranched sequences here. But again, familiar faces like N-acetyl uramic acid, but in different positions. So, I mean, Tony's mentioned sialic acid because he's an expert on sialic acid, and because his microphone is switched off, he can't comment on that. But this is one of these important monosaccharides based on the glycans of these bacteria. There's the acid group there, and here is the N-acetyl group, which is usually a hydrophobic part of the molecule, whereas the acid bit, of course, is hydrophilic. Now, there is an issue here because sialic acid is often the terminal saccharide in membrane glycoproteins, as shown here. See all the sialic acid residues. This is N-acetyl uramic acid is a type of sialic acid, and this is important because if you're generating a vaccine which is going to stimulate antibodies against sequences on the invading bacteria, then you have to be very careful that these sequences aren't also present on an individual's own cell surface, membrane proteins and glycoproteins and things, because the danger of that is you can generate a response not just against the invading organism, but also against cells, and we call that condition autoimmunity. So there is a danger which we have to be aware of causing autoimmunity when developing these types of vaccines. And these are some of the bacterial serotypes causing the disease of meningitis in infants. And it's not just Neisseria meningitis which cause these problems. Again, it's not all serotypes, but many of the serotypes. For example, there are over 100 strains of Streptococcus pneumoniae that have been identified and typed, but only or less than 20% cause serious disease, meningitis and pneumonia. So you have to bear that in with these things. But because several serotypes are responsible, that means you have to try and generate your response to be sort of polyvalent, to recognize the different serotypes, because you don't know which one is affecting. So you need polyvalent antibodies which have to be generated against all the dangerous serotypes, and this is a challenge to researchers both in the industrial companies working on this and also the academic university researchers. But this is only one of the challenges, and I've listed 4 here. One is effectiveness of the polysaccharide vaccine, and this is related to the type of response that the polysaccharide vaccine produces, the chemical purity of the polysaccharide vaccines, how reproducible the molecular weight distribution is in terms of the vaccines being produced, because we're dealing with polydispersed molecules here. And if you're a company making a particular vaccine, you have to be pretty specific to protect your patent, so that other companies know clearly what are the bounds of the particular vaccine. And this is where I was brought in years ago, when, and I can say this now because it's all gone and no longer confidential, was when GSK at Rixensart wanted to develop their own vaccines against meningitis and things, but Merck held the patents, and they asked me if, because we do a lot of characterizations, or have done, on these types of materials. We can characterize molecular weight distributions, things very accurately. Were the Merck patents absolutely clear in what their bounds were? And I had a look at them and I said, no, they're not. And on that basis, they were able to challenge the patent successfully. And that meant they could then go ahead and produce their own vaccine. So defined and reproducible molecular weight and distributions are very important, and also the stability of the preparation. How long do these preparations last for? Will a vaccine developed to protect, for example, infants here in UK and Europe be effective in Saudi Arabia or in India or in Africa? So stability is an issue, and there are flaws with polysaccharide vaccines. It gives generally poor protection, which is not long lasting, and that's because polysaccharides induce what we call an IgM response instead of a IgG or T cell dependent response. So it's T cell independent, and there's no memory effect. And we'll learn all about this when we do the antibody biotechnology module at the start of the next semester. So the industry, it was very clever actually what they did. They designed these conjugate polysaccharide vaccines. So you have your polysaccharide, which comes from the capsules of the dangerous bacteria, or sequences or part of the polysaccharide, and then you conjugate it to a protein which will stimulate the T cell response. So these are called conjugate polysaccharide vaccines, linking to an appropriate conjugate vaccine. Okay, so you take your polysaccharide and then you link it to the protein to give these conjugates, and there are set procedures for doing this. And obviously, you want your protein to have a conformation, so you can bind sufficient numbers of saccharide chains, something. So you want something to be somewhat elongated, ellipsoidal, as opposed to a compact sphere. And this is still an active area for I'll show you an example in a minute. So by conjugating to a protein, you can stimulate a T-dependent immunity, and this will enhance antibody production, and also because of the memory response. And by taking both boosters, you can give an increased response. So this is an active area for research and development, and in particular in terms of the type of protein which is conjugated to the saccharides. And there's 2 or 3 ones that are popular at the moment. One is the tetanus toxoid protein, not tetanus toxin, but it's been adjusted so it doesn't produce tetanus disease, so it's tetanus toxoid. This one, the Menactra one, was conjugated to diphtheria toxoid. So these are diphtheria and tetanus toxoids are very good at stimulating a T cell response. And there's a variant of diphtheria toxoid called CRM197, which is of interest at the moment, and that's what currently we're going to be doing with GSK. So if anyone's interested, we may have a project with GSK Rixensart, an MRes project characterizing the CRM197 protein and comparing it with the properties of the tetanus toxoid protein, which we looked at a few years ago and was published 2016, I think, 15. So we're going to be doing the same thing with the CRM197. So it is an active area of research. And this is just one example here. Menactra, it's quadrivalent. So it recognizes 4 serogroups, A, C, Y, and this one, W135. It's approved for persons over a wide range of age, and it's administered by intramuscular injection, not subcutaneous injection. Right, I don't want to eat too much into Tony's COVID stuff. So these are just some references which you can follow up with. The latest one with Thomas, who'll be involved with the project, and possibly Mary as well. So this is a good one to look at because it reviews what has been done before, but these other papers as well you'll also find quite useful. And with that,